welcome to The Art of Adventure. This is episode 268 with Mark Young. The Art of Adventure is the podcast that helps you travel the world, run your business, and embark on an epic quest. I'm your host, lead explorer and guide, Derek Loudermilk, and you can check out the show notes over at derekloudermilk.com. This episode is brought to you by Publishizer. Publishizer is the crowdfunding literary agency that I used to launch my last two books. And they've been super helpful with the whole process, from refining the idea to writing the proposal to funding the project, and then getting in front of traditional publishers, large international publishers, and the publishing contract, and basically the whole process. And probably I wouldn't have become an author if it wasn't for the Publishizer team. You know, they're telling me like, this is a good idea or that's a bad idea. and really giving me the push that I needed to go from just kicking around this idea of a book to actually writing. And lots of other authors that go through Publishizer find the same thing. So if you've been thinking about writing a book, then they have a contest starting August 1st. It's the Business Impact Book Proposal Contest. And this is for social media marketers, podcasters, coaches, speakers, women in leadership, activists, founders, trendsetters, educators, entertainers. Basically, if you're listening to this podcast, then you probably will fall into that category if you have a book idea. And it's going to be fun. You know, it's a contest, a little bit of gamification. You can win some money, and it's pretty simple. You go to publishizer.com/contests/2019/business impact, and you could probably search for Business Impact Contest on the Publishizer page. And you just go and you submit your idea for a book, and they will get in touch with you and help you get things started from there. So today's guest, Mark Young, was my academic advisor and mentor when I was a grad student at Montana State University. And he is a virologist, and he's known for discovering many new species in the hot springs of Yellowstone National Park. He's also on the board of the Society of Virology and many other universities and corporations and organizations. He's a distinguished lecturer, a fellow in the American Academy of Microbiology. His work has been cited over 15,000 times, and he's been on the cover of Nature and Science. So really important work that he's done. And recently, he has been the first person to create a synthetic virus, basically a new form of life entirely from scratch. So we're going to learn all kinds of amazing things, including the research that he does in Yellowstone National Park, which is why I picked him to be my mentor in the first place. He's super adventurous, unconventional scientist, loves to think outside the box with wild and crazy ideas, and also, you know, rigorous, highly qualified, one of the best scientists in his field. So you learn what it takes to succeed at the highest levels of academic research, how he comes up with his game-changing, wild and crazy ideas, and importantly, how to think about this brave new world of synthetic biology and creating new life and where we're headed in the life sciences and as humanity. So this is a fun, diverse, really interesting episode. I think you're going to love it. Here is Mark Young. Mark Young, welcome to the podcast.
Well, thanks. It's great to be here, and it's a pleasure to be talking with everyone today.
This is really exciting for me. For the listener, Mark was my academic advisor and mentor when I was at Montana State. And you were just telling me this story about going into Yellowstone to the park. In sort of, I think we'll be talking about the park because that's the local lingo. But I'm really curious, since you've been researching for decades now in Yellowstone, what are some of the most significant things that you've experienced in the park, or if you have any particular stories that you love to share from your research?
Well, I guess it's always good to start a little bit at the beginning and give a little context. You know, I came here in the early 90s. Actually, I came here to do mostly structural virology, and I've always been interested in the outdoors and what we can learn from nature. And obviously having Yellowstone really close at hand here was very intriguing to me. And I was kind of just on a whim, essentially. I was really curious about what was known about the viruses that replicate in these hot springs. So I did what any good scientist would do. I went to the library. In those days, you still went to the library, and tried to find papers from others about viruses in Yellowstone hot springs. And I was actually quite surprised to find that there were none, zero. No one had ever done it. And I was really found that curious because, Yellowstone was the first place that microbial cells, essentially at that time a bacteria, was isolated out of Yellowstone in the 1960s by people at MSU and other colleagues. And so, at that time, 40 years later, I was shocked that no one had ever looked for viruses. And the thinking was, my thinking at least, is anywhere there's life, there has to be viruses. The two are totally connected to each other. I don't think there can be life without viruses. And so, that started a quest. I couldn't get anyone in the lab at the time to go with me down to the park to look in the hot springs, and that started a process of going and figuring it out. And of course, no funding agencies were supporting that work at the time. It was totally driven by curiosity of what's there, and so that started the whole process.
And how long did it take before other people were interested, or you started getting financial support?
Well, so how the whole deal worked, and I won't say I was the first person in the world to think about this. So there was a professor at the Max Planck Institute in Germany, Wolfram Zelek, who had done something similar in Iceland. And so I jumped on a plane with a grad student, George Rice at the time, and we headed off to Germany to meet with Wolfram to learn whatever we could from him about his experiences of isolating viruses out of hot springs in Iceland. And he was quite helpful in pointing us in the right direction. And in fact, he ended up coming here to my lab for 6 months or a year, I can't remember, and we worked together. And that really helped. But it actually took us many years of working in Yellowstone before we were actually successful. So let me just give you an example. For any of your listeners, you've probably heard of a Petri plate, or even poured a Petri plate, which is basically an agar medium. It's a solid support that microbiologists commonly use to grow bacteria on. And, at the temperatures we're talking about and the acidic conditions that we're talking about, so we're talking about 180 degrees centigrade, 80 degrees Celsius, agar doesn't solidify at those temperatures. So it took us a while just to figure out how to pour Petri plates that we could use. And we had all these little things that we had to figure out along the way before we were successful. But, starting right around, 1998, 9, somewhere in there, we had our first indications that we had found viruses in these boiling acid hot springs, using Yellowstone as our laboratory. And what was really great about that was these viruses were unlike any viruses ever seen by science at the time, and that's exceedingly gratifying as a researcher to make those kinds of discoveries. And I guess you go to weird places and you find weird things, and that's exactly what we were hoping for, and what we did find.
And that's what essentially brought me to Montana State, is searching for weird things. Was there a moment when you realized you had discovered something? Like, were you looking in a microscope, or you were looking at a petri dish and you were like, oh my gosh, I think we have something? Was there a particular moment where you realized you had found something?
Yeah, I mean, I would say there was two big moments. The first was when you first see it with your eyes, and so how we look at viruses, as you know, is we can directly image them using an electron microscope, and the lab has several of these. So it's not something that the average lab has access to, but we have continuous access to EM abilities. And so we were concentrating samples out of the park, out of these hot springs, basically using filtering and centrifugation techniques to center in on molecules about the size of viruses, and then looking under the EM at them. And the first time we looked and saw these very odd-shaped objects, we knew we had something completely different. We weren't even sure they were viruses. It took us a little while to prove that to ourselves, that they were in fact viruses, because they looked so different than any viruses we'd ever physically seen before. And the second aha moment is when we actually got the first sequences back, the genomes of these viruses. And just like their physical image, their genetic signature was completely different and new, unlike any other viruses we ever seen. And though between those two things together, we were absolutely convinced that we were in uncharted territory, and that was really exciting for us.
And this is really exciting for myself and probably a lot of listeners. Just discovering something is really cool. But when sort of your average person asks about, why is this important? What do you say to them?
Well, first thing I say to anyone is thank you, especially if they're a taxpayer and, essentially are paying my salary, because I have the greatest job there is, which is the ability to ask fundamental questions about life, and to be privileged enough to be able to answer those. And, viruses to me are windows into life on this planet, and probably life anywhere else for that matter. And it's not just the discovery of these new viruses, it's what they tell us about life and how it evolved on this planet, and the role viruses play. And so, directly to your question about why should you care about viruses, and especially these weird new ones out of hot springs? And my answer is, you should care because they are providing new insights into the role viruses play on the evolution of life on this planet, and continue to play. And I think a lot of us are really interested in the big question of where are we going? Where's life going? How is it evolving? And our best way of figuring that out is to look about where we've been and how viruses have contributed to that evolutionary process. So just to give you some pertinent facts, viruses and people who have studied viruses have made some of the most important contributions we know about to life on understanding life. For example, the fact that DNA is a genetic material was first discovered with viruses. The genetic code was figured out in part by studying viruses. Fundamental aspects of how a cell works, including the processes of transcription and translation and DNA replication, was all figured out with viruses. And that's not just because virologists are really smart. That's because viruses are beautiful tools for asking these kinds of questions. And that's because viruses are perfect molecular parasites of the cells in which they replicate. What does that mean? That means viruses they have to mimic the biochemical environment in which they replicate. And therefore, they steal and they invent processes that are fundamental to how a cell operates. But a virus is much simpler than a cell. So it's much easier to first figure these things out by looking at viruses than it is by looking at the complex cell as a whole. And so that's what we were interested in studying these viruses from Yellowstone, was finding brand new fundamental aspects of life that we didn't know about using viruses as our probes.
And do you have a theory that you prefer about the beginnings of life? Well,
I've been, strongly influenced by others in virology and evolutionary thinking, and these go, back to Carl Woese, who discovered and reorganized life as we understand it today into three domains. I mean, I think many of us, if you're old enough, were taught, the five kingdoms of life, which turned out to be completely wrong. And there was kind of a quiet revolution that started in the late 1970s and really didn't take hold until the 90s, which was a three-domain tree of life with this new domain, archaea, being discovered. Okay, so we can talk about archaea and viruses and how that ties to Yellowstone in a minute, but back to your question of what's my favorite reason of evolution in viruses was the influence not only of Carl Woese, but of Eugene Kuna, who's written extensively about the role of viruses in early life. And basically, I am a firm believer that viruses and cells essentially had to co-evolve. So even before there were cells, there were protocells, as they're called, and there were protoviruses. And this had to occur for both the cell to emerge in life and eventually for viruses to emerge. And that there was this fundamental need in early evolution of life to exchange material between what would become cells and what became viruses, were these mechanisms for how to move genetic material around. And so virus-like things had to, in my opinion, exist at the very earliest stages of the development of life on this planet. And even today, we see that viruses play a dramatic role in driving and influencing evolution. So I see no reason not to believe that this hasn't been a continuous process all the way from the beginning of life on this planet.
And I just want to mention there's this amazing book by David Quammen, who previously has been a guest on the show, about Carl Woese and the sort of the untangling the tree of life and the sort of the revolution to reclassify and come up with the 3 domains and all that stuff. And thank you for sharing. I think that's fascinating, because when I would talk about the work that I was doing in Yellowstone, people have this image of the early planet Earth as sort of there's a lot of boiling water around, and so hot springs make sort of a great imagined corollary to that environment. And so I think the work that you're doing is almost like a time machine in a sense, that you're trying to look at very, very simple forms of life in conditions that are like the beginnings of the Earth. I mean, it just it's about as far back as you could sort of go right here, right now.
Yeah, I think one thing that I've really enjoyed about Yellowstone is the window it's provided for us on speculations about early life. And part of the argument is exactly what you brought up, which is there is evidence that at least early on, maybe not the earliest, but early on in life was probably hot life, thermal life. But clearly the life that's in these hot springs now is been evolved to be relatively recently to adapt to exactly the situation in that hot springs. And another way to think of the hot springs a lot is just how Darwin thought of the Galapagos, these islands, and these hot springs represent local islands of microbial evolution, where there's been local adaptation in each individual hot springs that we can follow and see how it's responding differently to the environment than another hot spring that may actually not be all that far away. So it really is this fantastic laboratory for asking questions about evolution.
And so Yellowstone is full of thousands, maybe tens, hundreds of thousands of hot springs of different types, salinities, pHs, etc. So it's just this weird place. Again, if we're looking for weird things, great location for that. And what is actually doing field work in Yellowstone National Park like, if you could summarize it?
Well, I think most people would think, gosh, you have the greatest job. You get to go tromp around the park as much as you want. And in reality, that's true. But when I'm recruiting graduate students to work on these projects in Yellowstone, I usually do it in the summer because it is beautiful and it's easy to get around. But I would have to remind people that Yellowstone is mostly, Montana in general, is a long, long winter. So it's a beautiful but tough environment to work in. There are estimated to be somewhere in the vicinity of 20,000 hot springs in the park. And we're really interested in a diversity of types of hot springs. It's that diversity is driven by the underlying geology of the whole system. So we visit these hot springs. The park requires that we work essentially out of sight of the public. So we are hiking or skiing into these sites, into the park. I've had some funny stories of prospective graduate students who told me that they knew how to ski. And when I say ski, I'm talking about backcountry Nordic skiing. And I just took them at their word that they were serious about that. And it's entertaining when, we head in the winter and I say, well, get your skis on, let's go. And they look at me with the blank stares saying, I thought you were kidding. And I'm not kidding. The backcountry of Yellowstone is absolutely breathtakingly beautiful. But actually to work in these sites, That's pretty physical, skiing in, carrying a lot of gear in, uh, taking measurements at these hot springs, uh, is a big physical effort, but quite enjoyable. Um, they're also very dangerous environments to work in. So I'm constantly worried about people getting hurt in these environments. So safety is a big concern.
Yeah, I remember thinking to myself, if I follow the bison tracks, if they haven't fallen through the crust, then I hopefully will not fall through the crust either.
Oh yeah, and I extend that to grad students. I usually let the first year grad students, the really big ones, go first,
There you
um,
go.
and then follow in their tracks. Just kidding. No, you have to view most of these thermal basins is a lot like doing glacier travel, where it's a crust that you're could break through, but instead of breaking through into a crevasse, you're going to break through into boiling acidic steam is incredibly dangerous. So we walk very carefully and are very cautious.
So you mentioned about recruiting grad students, and then you also mentioned sort of the other side where you sought out Wolfram Zillig. And in academia, there's this sort of institutionalized mentorship with academic advisors. I'm just curious if you could share your thoughts about how you approach seeking mentors and also being a mentor.
Well, let me broaden that a little bit. Let me talk about how to be successful in research, basic academic research. And maybe it extends to a lot of other areas too. There's really two ends of the spectrum, in my opinion. On one end, you do what everyone else is doing, and you do it really, really well. And you outcompete those individuals by just being better, smarter, and faster. And that's one end of the spectrum in science. The other end of the spectrum is you do things in science that no one else has thought about yet. And you're first to market, essentially. And you're successful when lots of other research groups jump into it later on. And all scientists, I think, are a blend somewhere along this spectrum of these two ends. And I would have to say, I'm probably much more towards being first to market and trying to do things and think about things that other people really haven't thought about that much. And I really enjoy that end and process of academic science. And that's where our success has been in those areas that we help start and create. And that's been very satisfying.
And could you talk a little bit about your process? So it's great to say, just do things that nobody else is doing, but you have to actually know where to look or think of the thoughts. So what's your process about that?
A couple things. And I don't know if I'm all that different than other scientists, but maybe a little bit, is I love taking in information way outside of my field constantly. When I was a kid, and my family, we'd go on family driving trips, essentially, around the West. I was a kid who was always looking out the window. And even today, I can still do this. And just look. I'm just fascinated at looking at almost anything. And so I want that input constantly. In my professional life, being at a university, I have this great opportunity where I get to interact with all kinds of people, and I purposely interact with people way outside of my field. And it's really a beauty of Montana State University is not such a huge university that I can't interact with physicists and chemists and musicians, all kinds of different people. And I need that broad spectrum of interaction, because that really informs my own science and how I think about things. And even, my outdoor activities, I find really informs me about science and just reading really broadly. I just want to know a lot about lots of different things, and it leads to questions of what if all the time. So the idea for me, coming up with questions and interesting things to do is not a problem at all. The real problem is what not to do, and where professionally the gamble will pay off scientifically in terms of new discoveries that makes the effort worth pursuing in the long run.
Can you give an example of something you've taken from a colleague in another field, or an idea that's triggered something in your virology work?
Oh, sure. I mean, I think one of the fun examples is actually work I started again right after I came to this university from another university, and at that time I was working on how viruses know how to assemble these beautiful virus particles. And the question was really to understand that in chemical and molecular terms. How does it know how to build these structures? And so I was heavily involved with detailed biophysical studies to figure that out. At the same time, I ran into this guy named Trevor Douglas, who later, for years, we have ended up working together. But it started with a conversation with him, and he's an inorganic chemist, and he was really interested essentially in how does an abalone know how to make a shell, and you're going, boy, what's the connection? And we ended up both over a squash game and a little red wine in my hot tub, talking about our respective interests at the time. And we came to this idea that we could merge these two things. and it's a beautiful moment of discovery. It was this idea that viruses, and think of a virus like a coffee cup in this case, is just a container. And that's what I brought, this idea of a molecular container of a virus. And what Trevor brought to the relationship was, anytime you have a container, you can do chemistry inside of it, just like cooking. And so we decided to see if that was really true. And again, no one in the lab would touch this project. So Trevor and I just did it ourselves. And we showed that you could chemically synthesize materials inside of a virus cage and make nanomaterials that were size and shape constrained by the inside of the cage. And this started a whole new area of nanotechnology, which really hadn't even been invented yet, and really exploded and was quite satisfying. Number of years of work in that area.
That's really a wonderful story. Thank you. And I just love hearing about, the sort of the friendly playing sports and sitting in a hot tub way that idea coalesced.
I mean, I would have to say, I find, people think scientists are, these geeks and nerds who just sit around in white coats and aren't socially interactive. But I actually find that effective scientists, are amazingly socially interactive. I learned so much by talking to other people, and I enjoy learning from other people so much. And and it really informs the way I think about science. And I love having friends that are, doing wild and crazy things way outside of science, because it helps me think about things in a more wild and crazy way, which is important for my job.
So a bunch of wild and crazy guys. Uh, you mentioned choosing to not do things, or there's so many interesting ideas you have to choose to not do some of them. Are there any things that you can think of that you turned down and maybe you regret it, or is there anything that you kind of wish you had done but didn't pursue?
Oh yeah, the most famous case is, and I think a lot of people have heard about it now, is this whole CRISPR field and CRISPR technology.
Mm-hmm.
So we were very involved with the CRISPR field before it was even known as the CRISPR field. And a student in my lab, I'm Blake Wiedenheft, and I became interested in these weird sequences that were in genomes, and then and at about the same time, a friend of mine came and did a sabbatical in my lab from the Netherlands, and he was interested in these odd sequences, and I remember having a conversation with him, and he said, "Mark, you gotta join me, and we gotta work on this.
Sounds like Darth Vader.
This is gonna be big, but it would have required me to." We were working on this system in one organism that was kind of hard to work with, and he was trying to convince me to do the exact same kind of studies, but in E. coli. And I basically said, no, I'm not going to dilute myself. I can't, Well, he went on to make some of the pivotal discoveries using the E. coli system, because it was the right system to choose for. And I won't say I ever regretted that decision, but, I would certainly be both rich and famous if I had heeded his advice at that time.
And I'm curious how you decided, or if it was an easy choice to stick to your guns, or sort of what that process was like for you.
Well, I think it's important to realize that, running a research lab is like running a small business. You're always having to make choices. And, I think people just think that money falls from trees to run a research lab, and that's not true. Anyone running a research lab is working their butt off to keep it funded. And what that normally means is to be attractive to funding agencies, federal funding agencies in U.S. science, which is, the National Science Foundations, NIH, NASA, all these different funding agencies. And that's an incredibly competitive world to operate in. And so you have to really be on your top of your game to get funded at these federal agencies. And you just can't switch fields right and left and be the expert instantaneously. So, I figure every kind of new project, whether it's finding viruses in Yellowstone hot springs or developing viruses for as molecular cooking pots, that took each one of those times at least 5 years of hard work with zero funding initially. That's high risk. And if it doesn't work out, you're in deep trouble as a research scientist. So, it's a lot of intuition about what is going to pay off, and a lot of sticking to it until you make those big fundamental discoveries that then attract funding agencies to support you. And likewise, it's hard to leave field sometimes when you're been highly successful and you know how to do it. But I've had to, purposely leave because other things became, exciting, and it's my job to always be doing something new. So I'm always trying to reinvent myself scientifically.
So I think this might be a good time to talk about synthetic viruses. You mentioned that that's something that you're thinking about and working on, and maybe you could just talk a little bit about what's interesting there, and before we get into potentially some of the ethical situations arising from that.
Sure. And so, this is a classic point where, I'm always thinking ahead for what's next. And what's next is, what am I really curious about? And so for a number of years in science in general, there's been this move towards creating a synthetic cell. And it kind of pissed me off because when you read about what people are really talking about doing, they're not creating a synthetic cell at all. They're basically taking current cells, minimizing them to the genetically speaking, so they're the smallest amount of information that still functions at a cell, or they're taking parts from different cells and mixing them together. But none of that's truly synthetic in my viewpoint. Synthetic means you create something that nature has never created. And so I was really intrigued by that possibility. And I said, oh, these guys are nuts to try to make a synthetic cell. It's way too hard. Let's try to make a synthetic virus. And they're much simpler. And so if we could make a synthetic virus, maybe that would teach us some rules about how to apply that or advance making a synthetic cell down the road. And, again, no one had ever done this or even thought about this as far as I can tell. And so I thought, over the years that we had learned enough about the fundamentals about how a virus works, both in chemical and cellular terms, to think we understood them. Maybe that's a little egotistical, but I thought we were there. So now let's see if we can, the idea was, now let's see if we can make one. And so I went off to do a sabbatical a couple of years ago in Europe, and I was going to do something else, but I decided, I only got one or two more shots at this, at something big. And I decided I'm going to start this project of making a synthetic virus. And it's not as crazy as it sounds, because what had really advanced is advances in computational chemistry mixing with biology. And there's this amazing guy in Seattle named David Baker, who was publishing a series of papers on computationally designing proteins completely de novo, so in silico. And proteins are the fundamental building block of function in a cell. And the idea was, could I team up with David and use his computational expertise to help design proteins that nature has never made that would make a shell of the virus? And that's the starting point. And unfortunately for me, David thought this was a great challenge. He's just an amazing individual. Really smart. I really like interacting with really smart people. I learn a lot, and I've learned a lot from him. So David and his students and postdocs computationally designed a series of proteins that would self-assemble into a shell. And he actually proved that this worked, which was amazing. So we then took some of his designs, working with him, and said, now can we make this a self-replicating system? And that's, if you know something about viruses, that's a lot about what a virus does. And so that's what we've been doing is linking these computationally designed proteins with the ability to replicate themselves, so they have a genetic material that replicates. And from our viewpoint, what's even more cool is we designed it so they would evolve, so we could watch them change. And again, back to my own theme of really interested in evolution and how does it work. And this was the ability, or has the potential to do the experiment I've always wanted to do, which is to start evolution at ground zero and watch what happens. And, no one's ever been able to do that, because everything we see today has been a function of billions of years of evolution. And by designing these de novo viruses, we're starting with something that has never evolved, and we're going to let it evolve. And we can start that experiment over and over again in the laboratory and ask, what happens? Does it always go down the same trajectory, or can it go down different trajectories? And so that's what's driving that whole intellectual curiosity is some of these fundamental questions. And so we're well on the way of creating these replicating systems, and we're quite excited about it.
That's fascinating. What about you said you designed it to evolve. So what does that mean exactly?
So, we know, actually it's a fundamental process of life, which is it has to evolve. And so viruses have to evolve. And so what evolve means is the genome has to change in response to changes in the environment, its chemical or physical environment. And that is what's passed on to the next generation. Okay. So we know a lot about the processes of evolution. How does that correspond? How does evolution change the genetic material, the DNA of a cell or of a virus? And we know that actually it's not DNA, but RNA has the ability to evolve much, much faster than DNA. And I wanted to do this experiment on a time scale of evolution that I could observe. And RNA evolution is, 1,000 to 10,000 times faster than DNA-based evolution. And so we designed this synthetic virus to have an RNA synthetic RNA genome that we can watch evolve.
Wonderful. Yeah. So I think it's probably important to note viruses can be DNA viruses or RNA viruses in there, and then also some variations in among those as well. So it seems like okay, you're the first person to do this, but the technology is there. So eventually, this is pretty interesting. Other people will start researching this, and then at some point, being able to create viruses, in let's say a decade or two, will be pretty easy to do, relatively ubiquitous for a trained scientist to do. At which point we run into the potential of somebody could synthesize a deadly human virus. What do we need to be mindful of, even in the face of such exciting potential to observe evolution? What are the risks, and what are the things to be aware of here?
Right. I mean, we are truly entering a new revolution in biology and life sciences, where we have gone through the process of discovering what evolution is several hundred years ago, to the process of mechanistically understanding how that works, But all evolution so far has been on random selection pressures that we can observe nature doing. We're now entering a phase in life sciences where we are leaving the world of what nature has done and going to the world what we can create. So we can do things and are entering in more and more things that nature could never do. And this is a brave new world. And as you point out, there is no question that this is going to have dramatic impacts, hopefully mostly for the good, but I guarantee you it has the potential also for unintended consequences, negative consequences, and even intended ones that people could eventually develop. And it's really going to, in the future, we're really going to enter a realm of really defining or redefining what even a human is, as we have the ability to change our own evolutionary trajectory by design. And these are huge ethical problems we're going to face. And, I think people have heard this before, but we've got a real problem in that our technology is well outstripping our intellectual abilities right now to deal with the ethics and to project the consequence of technology. And I think it's really important for everyone to be engaged in this debate and to figure out how do we continue to move forward, but in a responsible way.
And given that technology is moving so fast and these discussions sort of evolve at the social and cultural level, is it possible? And if so, who should be making rules about whether we're moving too fast? Because somebody might want to race ahead and somebody else might want to say, hold on, let's be more cautious. So who gets to decide?
Right. And we're, already being challenged by some of those difficulties in deciding who's going to be in charge. And this CRISPR technology is pushing that window, those discussions very quickly right now. So I don't have the answer to that. I think there's some clear things we should be doing a better job of. First, I think there needs to be as much transparency as possible. And this is a two-way street. I think scientists in general have an obligation to explain as much as possible exactly what they're doing, how they're doing, and why they're doing what they're doing to the general public. On the other hand, I think the general public also has an obligation to be scientifically literate. They have to, our education system has to provide enough information that these conversations can be had across a broad spectrum of society. Second, I think there has to be some level of trust, and I see this trust being eroded a little bit between science and society. We have to depend and we have to believe in, what scientists are saying, and scientists for their part have to be honest and open and upfront about both the benefits and any anticipated negative consequences of work they're doing. And so I think we can improve a lot. And I think we need, at times we need to put on brakes and we need to take a step back and reframe the questions and have a societal discussion. And this isn't new. I mean, we've done this in the past, and I think we we have to continue to do this in the future. And my favorite example is when, genetic engineering and cloning was first becoming possible in the mid 70s. Actually scientists as a group came together in what's called the Asilomar Accords and decided themselves to limit and slow down work in recombinant DNA technology until they understood it a little bit more. And they did. And that whole process worked. I'd like that as a model. I'd like to see more of that as we move forward. But with even a broader participation just beyond scientists.
And what is your personal or your laboratory's way of telling the public what you're doing besides coming on this podcast?
Well, I spend a lot of time engaging with the public if I can, either through direct conversations or through, documentaries, or anytime I have an opportunity, even when I'm on an airplane sitting next to someone who's, not a scientist and they have an interest, I'll be happy to talk to them about the kind of work we're doing and those kinds of things. I really want people to know what we're doing and why we're doing the work. So, constant communication, I think, is important. I think the scientific world, also, it's super important that we have open access publishing and journals that, allow everyone access to the latest scientific discoveries and findings. And they need to be written in a way that reaches virtually anyone. I tell my students, a good scientific paper should be written that your mom should be able to understand it, right? Someone who cares, but maybe is not versed in your particular field, should be able to completely understand what you're doing, how you did it, and why you're doing it. So I think that's super important.
Yeah, papers can be full of a lot of acronyms and I really appreciate that.
It doesn't have to be. And, at a certain level, you need to talk in the language of your field, but don't get lost in it. It's just like another language. When I travel in South America, I expect myself to speak a little bit of Spanish, at least to get around and to understand people. And you can do it if you're driven to be engaged. And I think that's important for all of us to remember.
What are your current concerns, or what do you think most about in terms of synthetic viruses and synthetic life?
Well, I think there's an obvious safety concern, and there's an obvious communication concern. So the safety concerns from a scientific viewpoint is pretty easy to deal with. We're at the very baby steps of this whole process, making a replicating virus. So this virus has no ability that we know of to spread. It can only replicate in a very weird and non-human background in cells. And so we think the safety concerns of it spreading to outside of the laboratory are nil. But we're always very cautious about that too. The communication, I'm really worried about people being afraid of this. And I don't want that to be the case. I want people to see it's just another step in our understanding of how life operates and how the process of evolution works, and that this could have lots of benefits, everything from developing new kinds of vaccines to advanced drug delivery systems. So I want them to see the benefits, and I want them to understand that we're trying to be really responsible in how we're approaching this, and that we're nowhere near any kind of level of creation that would have any that we can know of any negative consequences at this point.
And I can imagine that this is getting other people excited as well. Once someone sets the bar, people will get interested. Are you already starting to see colleagues and other virologists get interested, or what's the stage of the community as a whole right now?
Well, in general, I think there's a lot of excitement about about creating a synthetic cell. But again, we have a long, long way to go before that happens. We're still in the lab in terms of creating this synthetic virus. We have a few more experiments to go before we are confident ourselves that we've done exactly what we think we've accomplished. So we'll see how the scientific community responds to it. We're in conversation with bioethicists to try to get their insights into anything that we've not thought about yet. And those are fun conversations. But we don't know how the general scientific population and the general public is going to respond. We'll see after our first publications come out.
Well, it's really exciting, and I hope I like to have faith in people, and I think that if people are informed, then they can hopefully there won't be any sort of panic, and instead it'll be more of an enthusiastic response. But I'm sure I'm sure there are people who get scared or don't understand it, and I think that's a part of pushing the limits in any field. Really, there's going to be people who are for it or against it or don't understand it. So yeah, I'm glad that I know who's I'm glad that I know who's doing this research.
Yeah. And I think it's important to know that well before we started any of these experiments, we applied to an internal review panel at the university where totally independently other scientists and members of the academic community looked at the proposal to see if they could detect any possible problems with it. So this was all approved before we ever started any experiments. And of course, we keep a close eye on how it's all proceeding, and clearly, if we sense any problems, we'll immediately shut everything down. But so far, nothing has ever come close to raising that level of concern.
Yeah, that's good. I didn't know that there was that internal process, but it sounds similar to the genetic scientists agreeing to their own rules. I like the sort of self-policing that's going on here. Is there, broadly speaking, or about synthetic biology, is there anything that we haven't talked about yet that you think is important or that you'd like to share with the audience?
Well, I think it's important, again, that everyone tries to stay engaged and learn as much as they can about what's happening in life sciences nowadays. I think the challenges that people are going to be seeing and the change over the next number of years are going to be startling, I think, to many people. And we've got to engage in a conversation about these things. We are rapidly approaching times where the ability to manipulate humans is going to be or is a reality, and as a group we have to decide what are we going to allow and what are we not going to allow. And these conversations are going to be coming at us quickly. And I want people to be part of that process as much as possible.
Yeah. And I mean, there's conversations in artificial intelligence, there's conversations in life sciences. There's a lot of frontiers that we're leaping into all at once.
And they're merging, right? Nothing's operating in an island here. Life sciences and computational sciences and chemistry and physics and sociology, all are merging and impacting each other at an incredible rate, which is very exciting. But it's a brave new world.
I'm wondering if you think that it's actually possible for a person, an individual, to keep up, because if it is important for us to stay engaged and learn, but just the rate of new discoveries coming out, it's so fast. How can someone really keep up with things when they're changing so fast?
Well, you bring up an important part. It's impossible for any single individual to keep up with everything. Not going to happen. So I think it's important to have a very generalized background knowledge. That'll help, at least in the conversation. But we have to develop mechanisms where we trust people to be experts in certain areas and we listen to them. And trust is something that's earned. And we have to work very hard at gaining people's trust. In my own field, I have to work hard. And others have to work hard for me to trust them as experts. And how we develop trust is a really interesting process. And I'd like to myself learn more about how does, what is the psychology of trust and how can we make that better?
That's really interesting. A previous guest on the show thought a lot about that because he created this program called couch surfing where you can stay in strangers' homes around the world. And he's thought a lot about building trust and that led to the rise of things like Yelp and Uber and where you rate your interactions with people. Mm-hmm. So that'll be really interesting to sort of, if we can like universalize trust a little bit more.
Absolutely. Well, we're a long way from talking about the viruses in Yellowstone.
I know.
But it's all a continuum, and I, my own work, that is what I find so exciting is to be able to go out into a place like Yellowstone and to collect samples out of a hot spring and go all the way to ripping apart the atoms that make up these molecules that we pull out of a hot springs. And I just love that continuous spectrum. And I think that's, in the big conversation, that's what we're talking about in the changes of life science. It's a huge spectrum of change that is being thrown at all of us. And yeah, it's both fun and challenging at the same time.
Mark, I think this is probably a great place to wrap. Really, really appreciate you taking us through your work and all these great stories. Really, really fun to have you on the show. Much appreciated. Where can people learn more about what you're doing and find your work?
Well, if they need some bedtime reading, there's lots of papers that we publish. I guarantee it'll put most people to sleep. But I'm happy to talk to people. They can give me a call at work. I'll talk to them on the phone. Email still works, so they can contact me by email at myoung@montana.edu, and I'll try to get engaged in a conversation with them.
Wonderful. Best of luck on the rest of your work, and thanks again so much, Mark. It's been great to connect with you.
Well, it's been a pleasure talking with you. I love talking to former students. It's great to hear all the wonderful things you're doing. And if you want to continue the conversation at any time, just give me a call.
All right. Be prepared.
All right. We'll talk to you later.
All right. Bye, Mark.
Bye.
Amazing stuff. I mean, obviously, I got to spend time working with Mark, and there's so many things that I could pick out that he says in this interview, but the way that he approaches being an academic, exposing himself to a lot of ideas, trying to do things that no one's ever doing, and then also being very serious and rigorous before he leaps into something like synthetic biology. There's so many little things that you can point to say, okay, it's obvious that you have this level of success because you've done these things really, really well. So, and I'm really curious what your thoughts are about synthetic biology and how you feel when we talk about creating brand new life, even if it's something that doesn't infect humans. What is that trigger in you? If it does, are you excited about it? Are you scared? Would love to hear that feedback. Please feel free to email Mark, obviously, and message me or comment on my Instagram. That's one of the easiest ways to interact. And if you are the type of person that loves to be helpful to your friends, and you found this interesting, then they probably will too. So would love for you to share it with them, share it on your social media. And I think that we can all be better informed if we share these conversations with our leading thinkers and researchers and scientists. So if you want to subscribe to the show, then that also helps the show get out to more people. It helps boost the show up in the algorithm. If you leave a review, that's really helpful too. So if this is an important conversation that you think needs to get out there, then please do share it. And thank you so much for listening today. Now it's your turn to go out there and be adventurous.